Hello guys, how are you today? First of all, I want to tell you that this video today is going to be the last one in the part on the topic in biology, bioenergetics. And with this video, part 1, part 2, and this video, part 3, we will have covered all that I think is important to know regarding bioenergetics. Now, regarding the next topic, reproduction and inheritance, I have informed you that I won't be making videos right now. I will after the IMAT exam, but I don't want to give my time into making videos about this part of the IMAT, since there are so many videos online you can refer to, like many videos. I don't want to talk anymore about that. But after I have passed my IMAT, which is September 14th. By the way, good luck to everyone. After that, I will make videos for reproduction inheritance because I want to have the full set covered. And yes, then I will continue. The videos will be uploaded next will be about inheritance and environment, and last but very important, anatomy and physiology, which I can't wait to get into. Now, last but not least, before I begin about the part 3, photosynthesis, I want to thank you all for I want to thank you all for giving me your feedback, both in my Facebook account, for asking me questions. You see, you asking me questions, this is really helpful for me because I get to practice as well in questions, and by explaining them to you, I learn the topic better. So I'm glad to answer your questions. So don't hesitate to ask me, and for email me, always, guys. So I haven't logged in my mail all this time because I hadn't thought that anyone would have sent me an email, but turns out you guys have been sending me emails, and I'm sorry for not watching them earlier, and I answered to all of you. Okay, now let's get going. First of all, we're going to talk in this video about photosynthesis. Photosynthesis is the process by which light energy is converted to chemical bond energy and carbon is fixed into organic compounds. Anyway, blah 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 blah. We will explain all that later. The important I want you to know now is where photosynthesis takes place. It takes place inside the chloroplast. Of course, we have talked about the chloroplast in the cell part 2 video, I believe. Oh, that's my dog. Just give me a second, please. So what I was saying is that we have talked about chloroplast in the cell part 2 video. We have talked about cell organelles, and we have talked about its structure. I'm just going to make a quick revision and put that over here in order to remember everything. First of all, the chloroplast, like the mitochondrion, is made up of 2 membranes, an inner and an outer one, which together make up the chloroplast envelope. Now, there are these membrane stacks, layers of membranes. These membranes, which is layers of membranes, is called the thylakoid. All the stacks of thylakoids, they make up the grana, or in the singular form, a granum. Okay, and last but not least, we have the stroma, which is like the matrix of the chloroplast, which is the solution inside the inner membrane of the chloroplast. That's all I wanted to remind you, and let's get going into photosynthesis. What is photosynthesis? Photosynthesis is an energy transfer process. What it does, it transfers light energy into chemical potential energy for organic molecules. These can be then released for work in respiration. Now, Almost all the energy transferred to all the ATP molecules in all living organisms is derived from light energy used in photosynthesis by autotrophs. And we have talked in my previous video about autotrophs, what they are. But in this video, we're going to say we've got two types of autotrophs. We've got photoautotrophs and chemoautotrophs. The word chemo is derived from the word chemical or chemistry, so we got to do with some substances. And this is actually chemoautotrophs are organisms which do not depend on the light energy, but they use chemical energy sources. These include the nitrifying bacteria that are so important in the nitrogen cycle. What do? They obtain their energy from oxidizing ammonia to nitrate or nitrate to nitrate. Okay, and so that's all that we're going to say about chemoautotrophs and what they do. But now we'll talk about photoautotrophs, which they derived their energy from the light. Okay, they include green plants, the photosynthetic prokaryotes, and both single-celled and many-celled protoctists. Protoctists include the green, red, and brown algae. Okay, now what's happening in photosynthesis? Now I have forgot a very, very important word here. It's the trapping of carbon dioxide and its subsequent reduction to carbohydrate using hydrogen from water. And it takes place inside the chloroplast. But I have forgot a very, very important word, which is the word fixation. Let me write that for you in blue color. Trapping is not the official biological word. The official biological word is fixation. Let me write that down. Give me a second. Fixation. Okay. It's the trapping. Fixation is the trapping. Okay, it's like incorporating carbon dioxide inside other substances. So we've got carbon substances, organic carbon substances. All right. So when from now on we talk about fixation, we mean this trapping. Okay. Yes. Let me move on a bit. You see, an overall equation for photosynthesis in green plants is that we've got carbon dioxide plus water and light energy in the presence of chlorophyll. We have the conversion of a carbohydrate plus oxygen. But we have commonly formed hexose sugars and starch. So the following equation, this one, is commonly used, which is carbon dioxide plus water, a carbohydrate plus oxygen. Okay, now during photosynthesis, two sets of reactions are involved. These are the light dependent, like here, dependent reactions, and the light independent reactions. In the light dependent reactions, light energy is necessary, while in the light independent reactions, light energy is not needed. All right. Now, the light-dependent reactions only take place in the presence of suitable pigments that absorb certain wavelengths of light. Blah, blah, blah. What is that? We don't understand. I will explain it later. But what they do, this is the important thing. Why do we need light energy? It is important for photolysis. If you remembered by the last video, lysis comes from a Greek word, and it means the breakdown. So the photo means light. Photolysis is breaking down with the help of light. So what are we breaking down? Water. And what are we breaking it down into? Hydrogen and oxygen. And more specifically, electrons, protons, which is the hydrogen molecules, electrons and protons, plus oxygen. Oxygen is actually a waste product. Okay. Light energy is also needed to provide chemical energy in the form of ATP for the reduction of carbon dioxide to carbohydrate in the light-independent reactions. Now, we've got two photosynthetic pigments involved. The photosynthetic pigments, they fall into two categories: primary and accessory pigments. The pigments are arranged in light harvesting clusters called photosystems, of which there are two types, 1 and 2. Okay. Let me explain you this for a second, and then I will move on to explain it in much more detail. Here, what I'm showing you is that in a photosystem, several hundred accessory pigment molecules, which are two here, the accessory pigment molecules, surround a primary pigment molecule. Okay. And the energy of the light absorbed by the different pigments is passed to the primary pigments. So from the different pigments, the energy is being passed, the light energy is being passed to the primary pigment. The primary pigments are two forms of chlorophyll, and they absorb light energy and use it to provide energy to carry out photosynthesis. Okay. Now, let's talk a little more about light energy. You will listen to me talking about wavelengths, like absorbing and reflecting light and spectrum, and what on earth is that? Now we've got electromagnetic radiation, or waves. Waves, electromagnetic, are some complex waves. Actually, I can't draw right now, but I want to make them a simple wave in order to imagine it like that. I want you to imagine light right now as a wave. Alright? This is a wave. This is supposed to be a straight line, and this is a wave. This is the wavelength, which is, if you know some physics, you will know that this is the wavelength, how big or how long the wave is. This is a small, let's say, wavelength, it could be more. Whoa. This is a large wavelength. Okay. So according to the wavelength, we've got different types of radiation. If the wavelength is between 380 and 750 nanometers, 750 nanometers, then we can see it. If it is over that or below that, we cannot see it. The X-rays, for instance, have that much small wavelength. You see, between these numbers. But the visible light is between these. Now, a different wavelength, as you can see here, matches to a different color. Okay. So if we want to say like 680 here, is about 680, we can see that the color here is red. If I wanted to say like 430 here, we can see that the color is blue, and at 400 is purple. Alright, this is the spectrum of light, this right here. Now, where is it? Yes, here. Let me talk about that. Light is really intriguing, because what is light? Light is this. Actually, it's both a particle and a wave. But right now, because it helps me explain to you, I'm only looking at its wave nature. So light, we can see objects around us. You're all seeing objects. Objects are molecules together. And why can't we see them? I want you to go back into your physics, and let's say you're holding a book in front of you and you can see all types of colors, right? Why do you see these different colors? Are the electrons blue and red and yellow? What's going on? No. What's going on is actually that each molecule, each atom, it absorbs and it reflects some types of radiation. When we go into chemistry, I may be getting into more detail for that. But I want you to know that each molecule, that means object, they absorb and reflect radiation. And it's actually a complementary relationship, because what they don't absorb, they reflect. And what they don't reflect, they absorb. So let's say I have an object that it is green, that it is this color right here. Okay, why is it green? Because it absorbs all this radiation and it reflects that one. Because it reflects that one, my eyes catch this reflection and we see that object as green. The same things happen with plants. You see, we said that the pigment is chlorophyll. Chlorophyll absorbs mainly in the red and blue-violet regions of the spectrum. That means here, in the blue, here, this region, and in this region. Here is where chlorophyll absorbs. So this part, what does it? Reflects. And this is why plants are green. Okay. Now let's talk about the photosynthetic pigments. We've got, as we talked earlier, the primary pigments and the accessory pigments. Okay. The photosynthetic pigments of higher plants, they form two groups. The primary pigments are the chlorophylls, and the accessory pigments are called the carotenoids. Now, the chlorophylls, we can have chlorophyll A and chlorophyll B, okay, which absorb mainly in the red, yellow-green, and blue-green. Alright, while the carotenoids, we've got beta-carotene and xanthophyll. Now let me help you with that. Is that carot in, it has the word carrot in it, and carrot are orange. This how can you can remember this color. And xanthophil in the Greek language, the blonde is called xanthos. So the blonde is yellow, right? So xanthophil comes from the word xanthos, which is the blonde, is yellow. All right, I've talked about that. Oh, right, let's talk about that right now. Give me a second. Yes. Pigments found in red algae, the phycobilins, are reddish. They are reddish. Therefore, they reflect the red part of the spectrum. Therefore, what color are they absorbing? The blue and green range, which is here, the blue and the green range, because they are reddish. They reflect that color and they absorb that one. Okay. And they are called phycobilins. And now, chlorophyll B, the carotenoids, and the phycobilins are known as antenna pigments because they capture light in wavelengths other than those captured by chlorophyll alpha or A. Antenna pigments absorb photons of light and pass the energy along to chlorophyll A, which is directly involved in the transformation of light energy to sugars. And now we can see an absorption spectrum. What is? It's a graph which shows how much of the energy, the light energy, its substance, its pigment absorbs according to the different wavelength. You can see that chlorophyll B absorbs best at the blue and the red area. Okay, carotenoids, you can see that they absorb in this area, therefore in this area, the orange area, they do not absorb, therefore they must reflect it. Chlorophyll A in the blue and the red, therefore they leave the yellow and the green area. And this way you can make up this table, you can understand it with the help of this graph. And now we've got the action spectrum, which is again a graph of the rate of photosynthesis at different wavelengths of light. This shows the effectiveness of the different wavelengths, which is of course related to their absorption and to their energy content. Now, why does this show the effectiveness of the photosynthesis when we have the effectiveness of each one? So we see the effectiveness of photosynthesis at 650 nanometers wavelength. Why do we understand from here the effectiveness of the photosynthesis? Because the shorter the wavelength, the greater the energy it contains. So if we see that photosynthesis gains a lot of energy because it has a high percentage rate here in small wavelengths where chlorophyll A exists actually. Okay. And now let's move on at photosystems. What are the photosystems? They are light harvesting complexes in the thylakoid membranes of chloroplasts. There are a few hundred photosystems in each thylakoid. Each photosystem consists of a reaction center containing chlorophyll A and a region containing several hundred antenna pigment molecules that funnel energy into chlorophyll A. Okay, and we've got two types of photosystems, PS1 and PS2. What does PS stands for? Photosystem, therefore PS. Now PS1 absorbs light best in the 700 nanometers range, therefore it is called P700, while PS2 absorbs light best in the 680 nanometers range, hence it is called P68. P680, 680, okay. And PS2 operates first and then it is followed by PS1. Now let's start with the light dependent reactions. Light is absorbed by the photosystems PS2 and PS2 in the thylakoid membranes and electrons flow through electron transport chains. Alright, let's see now, we've got two possible routes for electron flow. But what I want you to know right now is that the light dependent, they include the splitting of water by photolysis to give hydrogen ions, protons, and the synthesis of ATP in photophosphorylation. The hydrogen ions combine with a carrier of mole NADP or NADP to make reduced NADP. ATP and reduced NADP are passed from the light-dependent to the light-independent reactions. Photophosphorylation of ADP to ATP can be cyclic or non-cyclic, depending on the pattern of electron flow in one or both types of photosystem. First, we're going to start with non-cyclic, which is the more difficult. And I'm putting these two steps. And we are going to read those steps in order to understand what's happening. But I'm going to go to our picture to see everything that's happening here. First of all, during non-cyclic photophosphorylation, electrons enter to ETC. ETC is electron transport chain, and the ATP and NADPH are formed. We've got the formation of NADP. Okay, we've talked about that in the past. Nicotinamide dinucleotide phosphate. This process begins in PS2 and proceeds through the following steps. First of all, energy is absorbed by the PS2 or P680. Electrons from the double bonds in the head of chlorophyll A become energized and move to a higher energy level. They are captured by a primary electron receptor. And then we have the photolysis. Water gets split apart, providing electrons to replace those lost from chlorophyll A in P680. Photolysis splits water into hydrogen and oxygen. Alright. Which is 2 electrons, 2 protons, and 1 oxygen atom. 2 oxygen atoms combine to form 1 oxygen molecule, which is released into the air as a waste product of photosynthesis. So this lives here. Then we've got the ETC. Electrons from PS2 pass along an electron transport chain to PS1. So they end up here. And what actually they pass is a PQ here, which is called a plastoquinone. A complex of 2 cytochromes and several other proteins, and they end up in PS1. This flow of electrons is exergonic and provides energy to produce ATP by chemiosmosis, the same way ATP is produced in the mitochondria. Now, because this ATP synthesis is powered by light, it is called photophosphorylation. Now, chemiosmosis. Protons that were released from water during photolysis are pumped by the thylakoid membrane. Okay. From the stroma into the thylakoid lamin. ATP is formed here. ADP is formed as these protons diffuse down the cradle from the thylakoid space through the ATP synthase channels and into the stroma. The ATP produced here provides the energy that powers the Calvin cycle, which is what happens next. Now we've got this one interesting thing happening here. NADP becomes reduced when it picks up the 2 protons that were released from water in P2. Newly formed NADPH carries hydrogen to the Calvin cycle to make sugar in the light-independent reactions. Energy is then absorbed by the P700. Electrons from the head of chlorophyll A become energized and are captured by a primary electron transport chain. This process is similar to the way it happens in P2. One difference is that the electrons that escape from chlorophyll A are replaced with electrons from photosystem II instead of from water. Another difference is that this electron transport chain contains ferredoxin and ends with the production of NADPH and not ATP. So what we have? We've got light. We get into the P2 and we got oxygen release. From here, due to that, we've got ATP produced. Then we've got the P1 and due to the P1 and the energy, we've got the NADPH produced, which carries electron, carries hydrogen. It carries hydrogen ions to the Calvin cycle. Okay, now let's move on. Let's see what's going on in the cyclic photophosphorylation. The sole purpose of cyclic photophosphorylation is to produce ATP. No NADPH is produced and no oxygen is released. The production of sugar that occurs during the Calvin cycle consumes an enormous amount of ATP. So periodically, the chloroplast runs low on ATP. When it does, the chloroplast carries out cyclic photophosphorylation to replenish the ATP levels. Cyclic electron flow takes photoexcited electrons on a short circuit pathway. Electrons travel from the P2 electron transport chain to P1 to a primary electron receptor and then back to the cytochrome complex in P2 electron transport chain. Cyclic photophosphorylation is shown here, and that's all. Okay. Let's move on to the Calvin cycle now. The Calvin cycle is the main business of the light-independent reaction. It is a cyclical process that produces a 3-carbon sugar. Here, it produces a 3-carbon sugar. Which is called PGAL, phosphoglyceraldehyde. Carbon enters the stomates of a leaf in the form of CO2 and becomes fixed or incorporated into a PGAL. We'll see later what's happening. Now let's see here. This process that occurs during the Calvin cycle is carbon fixation. So we take CO2 and we embed it. We do what? Carbon fixation. We put it in molecules. Okay. Now, next we have a reaction since carbon is gaining hydrogen. Now let's see. First of all, CO2 enters the Calvin cycle and becomes attached to a 5C sugar, 5-carbon sugar, which is called RUBP, ribulose biphosphate. And this way it forms a 6-carbon molecule. The 6-carbon molecule is unstable and immediately breaks down into 3-carbon molecules of 3-PGA or G3P here. It's called 3-phosphoglycerate. Okay, the enzyme that catalyzes this first step is called Rubisco, and I want you to know that. The Calvin cycle does not directly depend on light, instead it uses the product of light reactions, ADP and NADPH, and the Calvin cycle, like the light-dependent reactions, occurs only in the light. Yes, I'm sorry. Let me get you back to the Calvin cycle to tell you that you see here, we've got 1/6 of the molecules of G3P or triose phosphate, as it's called. And it is used to produce other molecules needed by the plant. Most of these triose phosphates condense to become hexose phosphates, which in turn are used to produce starch for storage, or I don't know, sucrose for translocation around the plant, or cellulose for making cell walls. Others are converted to glycerol and fatty acid to produce lipids for cellular membranes, or to acetyl coenzyme A for use in respiration, or in the production of amino acids for protein synthesis. Alright, that's all. Now let's go and talk about C3 plants. You see that in the light-independent stage of photosynthesis, the Calvin cycle, the carbon dioxide combines with RUBP to form a 6-carbon compound, which immediately splits into 2 3-carbon molecules. Let me show you that again. CO with RUBP, we've got 6, which is being split. Okay. These plants are called C3 plants. However, maize and sorghum plants, and most other tropical grasses, do something different. The first compound that is produced in the light-independent reaction contains 4 carbon atoms, and they are therefore called C4 plants. But why do tropical grass? This is C4 plants. But why do tropical grasses need to do something different from other plants in the light-independent stage of photosynthesis? The reason is a problem with the enzyme Rubisco. This enzyme catalyzes the reaction of carbon dioxide with RUBP. Okay, we've talked about that. But it also catalyzes the reaction of oxygen with RUBP, which is called photorespiration. It is an unwanted reaction because less photosynthesis takes place, because some of RUBP is being wasted and less is available to combine with carbon dioxide. Now this happens more readily in high light intensity, that is, conditions that are found at low altitudes in tropical parts of the world. But tropical grasses such as maize, sorghum, and sugarcane have evolved a method of avoiding photorespiration. They keep RUBP and Rubisco well away from high oxygen concentrations. Now, the cells that contain RUBP and Rubisco are arranged around the vascular, here are the vascular tissue, okay, the vascular bundles, and are called bundle sheath cells. These ones are bundle sheath cells. What happens here? The bundle sheath cells of the mesophyll cell. Carbon dioxide is absorbed by another group of cells, the mesophyll cells, which are in contact with air. So we've got CO2 gets in here. The mesophyll cells contain an enzyme called PEP carboxylase, which catalyzes the combination of carbon dioxide from the air with a 3-carbon substance called phosphoenol pyruvate, or PEP. This one. PEP plus CO2 give me oxaloacetate. Still inside, the oxaloacetate is converted to malate, which is then passed to the bundle sheath cells. Now the carbon dioxide is removed from the malate molecules and delivered to RUBP by Rubisco in the normal way. The light-independent reaction then proceeds as usual. Okay, I want to also to know that to ask you questions and take you back to the biochemistry, because by that we finished the today's material. Do you remember enzymes? You know, it's proteins, and we said that they have a range of temperatures which they can work, and they have an optimal temperature, which is the temperature at which the enzymes catalyzes the reaction in the highest rate. But how are in these cells that are so hot, how do enzymes exist and they are not becoming denatured? You see, enzymes have higher optimum temperatures. This is an adaptation to growing in hot climates. For example, in one study it was found that in amaranth, which is a C4 plant, the optimum temperature for the activity of PEP carboxylase is around 45 degrees Celsius. If the temperature drops to 15 degrees Celsius, the enzyme loses around 70% of its activity. By contrast, the same enzyme in peas, which are C3 plants, was found to have an optimum temperature at around 30% and could continue to work at much lower temperatures than in amaranth. Okay, that's all for today. I want to urge you to ask me your questions in the comments below, to send me an email. If you go to my YouTube channel description, you will see my email address there, or you can, if you have learned about these videos during the Facebook groups, then you know my Facebook and you can send me a message there. Thank you so much for watching. Bye bye guys.